Okay, so let's start the second topic. And let me briefly recap of a major program, the process of what happened in the cardiomyocytes when they contract. So let's go the forward direction. The uh, the action potential coming to a cardiomyocyte, they open up the memory calcium channel, and the influx of calcium ions directly go to a protein called RYR2 on the surface of SR inside the cardiomyocyte. And SR is a storage, like a bag, contain a high concentration of calcium ions. And then when RAR2 opens, the calcium ions, tons of calcium ions get out of the SR, get into the cytoplasm. And the calcium ions in the cytoplasm next will bind to the troponin, a small protein that anchor the tropomyosin onto the actin. So when the calcium ion binds to troponin, the troponin changes shape to remove the tropomyosin from the actin. So the myosin binding site on the surface of the actin will be exposed to the myosin, and then myosin gradually bind to the actin, drag them forward, causing the contraction of the cardiomyocytes. So this is how the muscle contraction, the mechanical part, or sometimes the chemical parts. But now we are going forward. What happens? What happened before? How does so this topic we mainly will talk about? How does the action potential eventually reaches to the cardiomyocyte? And originally, this action potential are generated by pacemaker cells. So before we even talk about this, so let's briefly review what is action potential and what is memory potential. And one thing first, do our cells carry any electrical charge? Anyone? Do we have actual any electrical charge ourselves?
Yes.
Yes.
Yes. Good question. I mean, good answer. So we do. All the cells have some level of electrical charge. And not only that, all of the cells are negatively charged. And the more the cell is active or responsive, the more negative that this cell will be charged. How does it happen? Why it happen? That's because on the cell membrane, there's always a pump. This pump called a sodium potassium ATP pump. Where do I have it? This here, sodium potassium pump. So in many cases, it's also called an ATP pump because each time it consume one ATP to pump three. You see that? One, two, three. Three sodium ions outside the cells, and then a pump two potassium ions inside the cells. In this case, as you can see, three out, only two in. Given time, gradually the cell will accumulate a lot of a negative charge inside the cells, and and, and gradually this cell carry a negative charge. And this negative charge, even though this pump is always actively working to pump sodium out, this pump is not under regulation, under active regulation. In most cases, this pump just uh, all constantly active, and then they constantly pumping the sodium out and potassium in. However, that's the thing is, so then even though this pump always working, but at a certain level, when the cell memory potential reach in a certain low negative level, It does not further go into more and more negative. There's a balance. This balance is determined by that. Even though in the last semester, maybe you have learned that the ions cannot pass the cell memory by itself. But actually, that's not absolutely cannot happen at all. There's always a leakage, especially for the sodium and potassium and small ions. There's always a leakage of the sodium and potassium ions across the cell membrane. So in these cases, when you have pumped tons of sodium out, some of those, the more you pump out, the stronger the sodium ion will migrate back to the cells. And also, the less even the less sodium ion inside the cells and the more potassium ion inside the cells, and making it much harder for the pump to keep doing this work. Just imagine you push some stones further and further up a hill. At a certain level, you no longer have this capacity of pushing further up. And then how far away you can push the stone uphill. It depends on how many people you have. In some cells, like a cardiomyocytes, like a neuron, and like a skeletal muscle, on their surface, they have a lot of sodium, potassium pumps on the surface, so that they can push their memory in a very high level. I mean, actually, it's sometimes confusing here because it's negative, but still is a high negative level, I would say, into a very high negative level here. But in some other cells, like one of the most uh, inactive cells in our body is the cartilage cells, the chondrocytes. They are very inactive. So their memory potential, you can take a look here, is very small, only -8 compared to -90, 80 in the cardiomyocytes, in the skeletal muscle cells and the neurons. So that's why I guess maybe it happened to you. For example, on a weekend day, Saturday or Sunday, you really do not want to do anything. You just lay there on your bed, do nothing, not even watch your cell phone, but still you consume a lot of energy. And more than that, if you just doing nothing together with your friends, even though boys and girls both lay down for the whole day doing nothing, still the men still have to eat more food than the women. That's because the man has more muscle, and the muscle, even though the muscle do not really do any work, it still have to consume a lot of energy just to maintain this very highly negative memory potential. And this stable memory potential, we call it the resting potential. So I talk about a lot, but this part I don't think it's in the test, but I need to give this as a background information in case you do not remember what you learned from last semester. So then I hope now you know what is the memory potential. And then what happened, since now you have the memory potential, what happened if the cell is activated? When the cell is activated, this is the memory potential. This is the actual potential of a neuron. This is the actual potential of cardiomyocytes. In both case, this when the cell need to be activated, this memory potential dramatically, rapidly going way up to the top. It used to be a negative, and this status, when a cell have a negative memory potential, this status we call it call it polarization. And then when this cell is activated, this memory potential dramatically going the opposite direction from negative to positive, and then so positive even past a zero, going all the way to a positive to a positive to a positive actual memory potential. And this process we call it depolarization. And here in the last semester, if you remember, when we talk about the neurons, there is a complicated open of the ligand gated sodium ion channels and voltage gated sodium ion channels. And the sodium ion channel closed, now you have the open of the voltage gated potassium channels. It's in all of those, we will not be testing this semester. It'd be great if you still remember, but I do not really have time to go over all of those. But in this semester, it'll be great if you can remember how does the membrane potential go in the during when this cell is activated. And this action of the membrane potential from polarization to depolarization, from negative to positive, and then to negative again, and sometimes even more negative than before. This whole process of the actual whole change of the membrane potential, we call it action potential. So this how it changes. And then the action potential chart is totally different between a neuron and a cardiomyocyte. This is a neuron. Resting potential going up, depolarization, and going down, repolarization. And the repolarization even going even further, more negative than it before. And they call hyperpolarization, going down and going back again, eventually back to the resting potential. This is a typical neuron action potential. And this is a typical of the cardiomyocyte action potential. It's also going all the way up. This is the depolarization. This is the resting potential of the cardiomyocytes, and depolarization going all the way up to a positive action potential. And then the key feature about cardiomyocytes that you have this plateau phase for a very long time. Do I have it? No. Oh, I measure this later. What's happening here? We'll talk about this again, but let's say this. This after the peak positive membrane potential, there's a relatively long time when the membrane potential actually does not change a lot. And this is the feature of the cardiomyocytes. And this is called a plateau phase of cardiomyocytes. We will talk about this later. We'll talk about it later again. And then let's focus on another thing that we have talked about before in the last semester. How the action potential can say this, how the neural signals or the other activation signals can be transmitted from one place to another. And in the case of a neuron, think about this. We touch something. I touch here. I can feel it. I touch the other finger of mine. And in my fingertip, there is a receptor can receive the mechanical pressure. But this signal has to be transmitted all the way from my finger into near to my spine. And this signal has all the way transmitted to my brain. And in the case of neurons, how does this neural signals being transmitted from one place to another faraway places? And in the case of the neurons, this we call action potential propagation. How does it happen? Here I'm showing an axon of a neuron. Whenever one place have an action potential, tons of potassium ions get into inside the cells into the axon, and then this sodium ions, based on a physical law, they do not stay here just still. They want to diffuse to the next region, and when this sodium ion diffuse to the next region, they increase the membrane potential, make the membrane potential actually less negative. When this less negative reaches a certain level, it passed the threshold membrane potential. I hope you still remember threshold membrane potential. And then once it passed the threshold membrane potential, the voltage gated sodium ion channel opens. And when this opens, it triggers a depolarization of the next region. And so the action potential will happen to the next region. And when the action potential happens to the next region, sodium ion get in again. And this sodium ion will further diffuse to the next region. And then it triggers the action potential in the next region. In these cases, this action potential has just happened one place, another place, another place, another place, another place. They call it action potential propagation. And for the action potential propagation to happen, the key thing is that the sodium ion can pass from one place, can diffuse from one place to the next place. In the case of a neuron, it's easy to happen because a neuron, a single cell has a super long axon and is always within the one single cell. And in the case of a skeletal muscle, it also can happen because if you remember, we compared the skeletal muscle to the cardiac muscle. In the case of skeletal muscle, all the cardiomyocytes fuse into one single fiber. And then there's no membrane to separate intersections within one muscle fiber. In the case of a muscle fiber activation, normally a nerve ending activate the action potential in the center of the muscle fiber, and then this action potential just migrate through this action potential propagation to the end, all the way to the end of the muscle fiber. Next, most important thing here, none of those will be tested, but the only thing can be in the test here in this semester is how does the action potential can propagate from one cardiomyocyte to the next cardiomyocyte. Between two cardiomyocytes, the two cardiomyocytes are connected by this structure called an intercalated disc. In the last topic, we mentioned there are two types of cell junctions there. The desmosome and the adherence junction. The desmosome is the anchor to physically anchor each single cells together to resist the mechanical shear force. And the adherence junction is to transmit the mechanical force from one cell to another. And the third one, we call it gap junction. The function of the gap junction is that it's basically a tunnel. And through the tunnel, the sodium ion can pass freely across. In these cases, the sodium ion can diffuse from one cell to the next cell. When the sodium ion can pass across the intercalated disc, then the action potential can propagate from one cell to the next cell. I repeat it. The key words, key sentence of this topic is that the action potential can propagate from one cardiomyocyte to the next cardiomyocyte because the sodium ions can diffuse across or through the gap junction between two cells. Okay. Now let's further move on. So action potential. How does action potential initially happens? Where you do have the action potential happen. And the thing is, you don't really want action potential to happen out of nowhere. And in any cases, if action potential happen out of nowhere, there's in many cases there's a neurology disease. In some people have this chronic pain coming out from nowhere. That's one of the disorders that action potential just generated without even a stimulation. And it's also some people, they say they hear things, see things that not really happen. There's also something like that. But it's more complicated, but they're similar. So the thing is, now we trace back to see where does the action potential first, where is the first place that the action potential is generated. And before we use this example, I use this finger to touch another finger. I can feel this touching. How do I feel the touching? That's because there's a mechanical receptor here. And this mechanical receptor we call piezo. This is a very famous protein. It won a Nobel Prize, I think 5 or 6 years ago. No, not that long. Maybe 3 or 4 years ago. But anyway, this is a kind of a ion channel on the surface of the cells. It's in a normal status, resting status, something like this. It's inward located in the cell membrane. And just imagine if something pushed down here, it pushed and then it used to be down inside inward in the cell membrane, and the other places it's being pushed. The cell membrane is being pushed. It going up and open, and when it's going up and open, calcium get in, and the sodium get in. It triggered a depolarization. That's what happened That's what happened in the stimulus touch stimulated action potential generation. This is where the action potential is first generated in the touching. And however, okay, yeah, I just say something. I just say something that if stimulus can happen out of nowhere is a bad thing. I mean, I have to take it back. In most cases, if a stimulus happens out of nowhere, it's a bad thing in the nervous system. But there's almost only one case in our body that we do need this action potential to happen out of nowhere. Actually, not really out of nowhere, but in the let me see. In the heart, there's a special cardiomyocytes called pacemaker cells. These are pacemaker cells. Again, these pacemaker cells, their major function is to generate the action potential. This is where the action potential is first generated. But these cells, they are not neuron cells. They generate action potential, but they are not neuron cells. They are specialized cardiomyocytes. And they are so special, they do not even have any sarcomere for contraction. Rather, they mostly specialize in just generating action potential. But how do they generate the action potential? This is the action potential of pacemaker cells. One of the features here is compare this one to a cardiomyocyte or to something else we talk about, like this one. This is the neuron. This is the resting potential. This is a resting potential. Resting potential is a straight baseline that stays there for long. But look at this. Look at the resting potential of the pacemaker cells. A pacemaker cell does not have a resting potential. It does not have the straight baseline. Why so? That's because here you have the action potential of the pacemaker cells. Let's say for some reason a depolarize and repolarize, and then when they're going further negative, and then reaching a certain point right here, it does not stay there. Something happens at a certain point. Something happens that making the memory potential of the pacemaker cells going up again. What happened here? That's a key thing here. There are several proteins named you are required to remember for this semester, and this one is one of those. The full name is hyperpolarization activated cyclic nucleotide gated channel. It's a long word, but the word make sense. Let's first focus on the first two. Hyperpolarization activated. The H. H means hyperpolarized activated. N means, what is N? Nucleus, gated, whatever. Yeah. What do you mean by hyperpolarization activated? Coming back here. Hyperpolarization is this one, when it's going all the way down. And then when they reach to the bottom, so this channel is open. This is an ion channel. And this is open. This is open when the cell memory reach a low level. So actually, when the scientists first discovered this channel, they have no idea what's the function of this, because all of the other channel they have found are activated by some certain level of a uh, high level of cell memory potential. But this is only one they have found that when the memory potential reduced to a certain negative level, it's open. And since they do not really know the name of the function of this, actually given a name called it funny ion channel. And then this so-called funny ion channel, actually that's the fundamental mechanism of how the pacemaker cells can spontaneously generate a rhythmatic action potential. And that's the fundamental way that how we can survive. We cannot survive if the pacemaker cell do not generate a signal. And because if there's no such signal, the heart cannot beat. So there's a one feature of, again, let me repeat one feature about HCN channel. In the one feature about HCN channel is that it's only open when the cell memory reach to a certain negative level, going all the way down and it's open. Because of this, now you do not really have the pacemaker cell do not really have any resting memory potential. Rather, it going up and down. Because when going up, there's another type of channels that close or open in the higher memory potential. And then when going down, another channels have a lower memory potential. This is like, imagine now you have two people play tennis. The balls is being hit this way, that way, this way, that way. In this cases, now the memory potential just have a fluctuation up and down, up and down, up and down. And whenever it's going up, it generates an action potential to be passed to the next cells. And that's one thing that we talk about, that how does the how how can the HCN channel can open by itself. And next we talk about another thing. Our heart rate can be someone. Susan, raise your hand. Anything? No?
Yeah, I was copying something from the other slide. You changed the slide, right?
I don't remember, did I? I don't think so.
I think, yeah, I think you just changed the slide. So I was copying something and all of a sudden it wasn't there.
I didn't. I don't think so. Where would you go get a slice?
Yeah, I think it was on the propagation, was it on? And you changed it back to the action potential? Yeah, something like that. I don't, God damn it, I forgot which one it was on.
So yeah. So we can further discuss later. I'm not sure which part you're talking about.
Yeah, it's
But
okay. It's fine.
I
Sorry.
believe, yeah, if you realize the one that in the contents is not the same as this one, please let me know. I'll update that one. But I don't think so, because I don't really change anything for this summer. Okay, let's continue. So on Monday we talk about the heart rate. Heart rate can be regulated, and how does the heart rate be regulated? So now we already have this, like say, ping pong ball coming up and down, because there are multiple channels. What it, the multiple channels open and close based on the cell membrane. And then, so what kind of things that determine the rhythm? And in this cases, you can compare this one and this one, this one. You can see that once above the threshold, the shape of the actual potential actually doesn't change much. And this is a regular normal heart rate. And this is a slow heart rate, a low heart rate, and then a high heart rate. And then what happened here is that which part is changing? This part, this part. this part changing. So the really changing time is that after the HCN channel is open, how long it take for the membrane potential to elevate all the way to a threshold membrane potential, and why this one take much longer and this one take shorter. We have to coming back this again. So this HCN channel, it can open. It's not but it's not like some of those ion channels. Probably in your home there's some light. Some light is on and off, on and off. There's no adjustment. But some light you can turn it brighter, you can turn it dimmer, and for most other ion channels in the neuron or in the muscle, they are just on and off. But HCN, it is tunable. You can make it wider, you can make it narrower. When you can make it wider, when you make it wider, and then this the sodium ions will get into the pacemaker cell much faster than normal, and then you will reach a threshold much faster than normal. And then in this cases, the time interval between two action potential become much shorter. And then in these cases, you have a much higher heart rate in these cases. And then on the opposite, if the HCN channel is only open a narrow space, now the high sodium ion can only pass slowly through the HCN channel, and then it takes much longer for this pacemaker cells to reach the threshold. And then in this cases, the time intervals between two heartbeat will be much longer. And then in this cases, this guy now have a much lower heart rate. So now we know that the heart rate actually is determined by how widely that is HCN channel is open. Then the next question is, what determines that how widely this HCN channel is open? Let's coming back to the name of HCN. Hyperpolarization activated. Now we know this name. Hyperpolarization means it's activated when the membrane potential is reached a very low, very negative level. And what does it mean by this cyclic nucleotide gated? What is cyclic nucleotide? Actually, you don't really have to know this name, but the abbreviation called is CAMP. CAMP is a very widely used messenger signals, and in these cases, the high CAMP will bind to some proteins that drag the channel open wider, and the low CAMP will actually say this way: low CAMP is passively making in this. Let's say this one: if there's no CAMP, then the HCN just open by itself is only very narrow. I mean, almost cannot open at all. So the more CMP bind to this another protein that bind to the HCN channels, the wider the HCN channel will be open. So in other words, CAMP can cyclic AMP can quantitatively determine the diameter of the HCN channel. More CNP, wider open of the HCN channel. And then wider open of the HCN channel, faster heartbeat. Next question. What kind of conditions determine the CNP? And actually in the last class we talked about it. What's the directly condition that can actively that can actuate a faster heart rate, a higher heart rate? Sympathetic nerve signals. So sympathetic make heartbeat faster. Parasympathetic make heartbeat slower. Why? Now we have the full circle now. We have closed circle now. When the sympathetic nerves activate to the pacemaker cells, it led the pacemaker cell to release more CMP. and more CNP bind to another protein that control the CN to control this HCN channel to make the HCN channel wider, and the wider HCN channel will make the a wider CN will make the heart rate higher, faster, and then the parasympathetic will inhibit the CNP released, and then the HCN channel will be narrower, and then the heart rate will be slower. That's how we. That's basically how that people, well, we regulate our heart rate. And then now let's go to a level up. Now we just before we just based on one single pacemaker cells, and of course, in the heart there's not only one pacemaker cells. There are, I don't really know the number, honestly. Maybe tens of thousands, not that many. There's the pacemaker cell, the ratio of pacemaker cell to cardiomyocytes. I think I have the ratio somewhere. Oh, here, got it. So there's only about 10,000 pacemaker cells, and then you have 1 billion cardiomyocytes. So very few pacemaker cells, but 10,000. But all those 10,000 pacemaker cells, they all join together, and they connect together to form this. We call it the conductive system of the heart. And we already mentioned a little bit in our first class, and then today we talk about furthermore in details in this conductive system of the heart. In general, I mean, there are two nodes here, AV nodes and SA nodes and AV nodes. SA nodes is on the top of the right atrium, and AV nodes is between the right atrium and the right ventricle. And from the SA nodes send out mainly 4 major branches, and these 3 of those going all the way down to the AV nodes, and 1 node go all the way to the left atrium, called it Bachmann's bundles. And these major tracks or bundles, all of those, not only they connected to from SA nodes to the AV nodes, but also they reach out the small fibers. At the end of this conduction system, there's one cardiomyocytes at the end. So this is more like a tree. You think about this tree has 2 roots. One is SA nodes, the other is AV nodes. And from the tree at the beginning, there's branches, and the pacemaker cell join together as the branches. And after these major branches, now we have the small, and it gradually have the small branches. Those small branches are the cardiomyocytes, and more cardiomyocytes. So there's not. They say not every single cardiomyocyte directly connect to pacemaker cells, and because after this first cardiomyocytes, the cardiomyocytes next connect to each other through the intercalated disc. But overall, just imagine 2 big tree. A big tree have 2 roots. One is SA, another is AV nodes. And then the thing is, the end of each of those ends, like leaves, the distance. There's let's say, yeah, it's big. They have two roots, and each root have 1 big trunk here. One is SA and one is AV. And then this in its case SA nodes, it send out the fibers to all of those cardiomyocytes in the atrium, into atrium. And then the thing is, this conductive system is so well wired that the time that it take for the action potential to be transmitted from the SA nodes to the end are exactly the same as each single branches. So in these cases, that's why all the cardiomyocytes can contract together at the same time. That's the cardiac cycles we talked about in the very first topic. The 2 atrium contract together, and then at the same time, the SA node also send a signal to the AV node, and this taken about 0.1 second to send it from SA node to the AV node. After the AV node, it passed through the His bundle, and eventually the Purkinje fiber. The same thing. From the AV node, another big branch going out, and several branches. Eventually, the thing is, again, it's so well wired. All of those ends of these three branches receive the signal at the same time from the AV nodes. In these cases, they all contract together. That's the beauty. That's the beauty of the conductive system of the heart. And so there's another thing, but in the first class we talk about the key feature about a heart is to make sure that every single cardiomyocyte in the ventricle can contract together. How we can do so? One of the approach we just mentioned, is that the distance between each not really each single, but each new branch, each local cardiomyocyte to the AV nodes, a distance. They receive the signal from the AV nodes almost at the same time. I say almost, but not exactly at the same time. That's because the thing is, not every single cardiomyocyte are directly connected to a pacemaker cell. Normally, there's only one cardiomyocyte connected to the pacemaker cells, and the rest of those are actually connected to the first cardiomyocytes. So inevitably, the first cardiomyocytes receive the signal first, and then next, there's still a tiny time intervals difference between several sequential cardiomyocytes. How do the heart solve this issue? Let's coming back to this. This is the cardiomyocytes action potential. Particularly ventricular cardiomyocytes. It has a very long stage called a plateau phase of action potential. So why they have this? Mechanically, in a molecular level, at this moment, both potassium channel open and the calcium open. Potassium get out of the cells and calcium get in. Both potassium and calcium are cations, are positive ions. So one in, one out. Actually, it remains a dynamic balance of the cell membrane potential for a short period of time. In these cases, there's no change of the action potential. Since there's no change in action potential, this calcium keep open. And when this calcium keep open, this calcium channel on the cell membrane keep open. And then a RBR2 keep open. And then this cardiomyocytes keep contract for a relatively long time. So in these cases, even though the first cardiomyocytes, the second cardiomyocytes, and the third cardiomyocytes, even though they do not start from the same time, however, because each of those have a prolonged contraction time, so they have an overlap. Each of those have an overlap. So in the whole heart, there's certain time, because we have elongated, we have expanded the time that the heart can contract. So we can find a long overlay at which time that all the cardiomyocytes is contracting. So in this case, let's repeat how the cardiomyocytes can contract at the same time in the ventricle. First is that the signal take roughly almost the same time from the AV node through the Purkinje fiber to all of those local regions. And secondly, even though there's a tiny difference in the contraction time, because the longer plateau of contraction, there's always an overlap between the contraction of the cardiomyocytes. Any question so far?
Yes, I have a question.
Yes, please. Go ahead.
So somebody who is on beta blocker because they had a heart attack,
yes,
does that mean that the HCN channels doesn't open?
beta blocker. That's a very good question. Beta blocker is a drug he used a long time and still the primary drug. So what's the really function of beta blocker? Beta blocker does not really the target beta blocker is not HCN channel. That's a very good question. I was thinking about whether to talk about beta blocker. Beta blocker is the suppressor of sympathetic nervous system. Does it make sense?
Yes, but I understand because when that's why I asked a question, because
Yes.
when somebody's on sympathetic, that's when they release epinephrine, which is increase the CAMP. So if the epinephrine is black, that means the CAMP will not increase.
Yes. So it's a suppress the sympathetic nervous system, and so that if sympathetic nervous system is not activated, there's less CMP, and then the HCN channel is not wide open. Actually, I will say this: HCN channel is not a direct target of the beta blocker, but beta blocker indirectly. I mean, not indirectly. It's more downstream of the beta blocker. The direct target beta blocker is the sympathetic nervous system.
Okay, so in other words, like somebody on a beta blocker does not fight. The body does not prepare for stress. They cannot fight stress.
No. Oh, that's a good question. That's a good question. I would say I'm thinking they can still. So basically here, their overall sympathetic nervous system is reduced, but still they can be activated, but not as high as it's supposed to be. So they can still be getting excited. And but in the case, so let me actually, there's a little bit complicated question now. Let's say, what's the function of a beta blocker? Beta blocker is normally used to given to people that had a heart attack, but I just realized there's people there was say a misunderstanding of the function of the beta blocker. The consequence of the heart attack cannot be treated by beta blocker. What's the function of a beta blocker is to prevent another heart attack. So how beta blocker to prevent another heart attack? To prevent the body from getting too excited. So overall, it constantly suppresses the sympathetic nervous system. However, this sympathetic nervous system still can sometimes going up and sometimes going down. But overall, compare the same person with or without beta blocker. This sympathetic, the peak of their sympathetic nerves actually much lower if they take the beta blocker. So in other words, a guy taking beta blocker, it can efficient but prevent this the active fight and fight or flight response of the body. I hope. Does it answer your question?
Yes, thank you.
So welcome. Thank you for the question. So let me see. We are almost finished the most important part of this class, and then so in here we just we are going to have a lab focused on this one. We'll talk about more details about this, and in general, here let's try to read the ECG, and probably I guess many of you saw this before. And there's a general ECG or EKG. ECG is English, EKG is Germany. But if you go into the hospital, actually EKG is used more often than ECG. I mean, I don't know why. But anyway, so what is this? The ECG and EKG, there's a common misunderstanding is that these are the signals from pacemaker cells, which is wrong. Pacemaker cells, even though pacemaker cells generate a signal at the first time, but Just remember the date. Remember the number. Pacemaker cells has only 10,000, and cardiomyocytes has about 1 billion. So this is like we have leaders and we have the politicians. They can give us the idea. They can sometimes, if they're good, they can lead us. But what real power is in the human people, the big population. So the ECG or EKG is the overall, in general, electrical behavior of the heart. So basically, how we do the EKG, we compare the heart electron charge with, in most cases, say the right arm here, this one here to here, and then the majority of the electrical feature of the heart is actually contributed by not by pacemaker cells, but by the cardiomyocytes because they are huge. They are huge in number. And then this is basically eventually what the exam, what this machine showing the EKG is the combination of all of those action potential of the, say, for example, the atrium cardiomyocytes, ventricular cardiomyocytes, AV nodes pacemaker cells, SA nodes pacemaker cells, all of them together. However, which one contribute the most? Again, cardiomyocytes. And that's why the highest peak, this R peak, this whole thing called a QRS complex, and the peak, and this is called a wave, not peak. The wave, the R wave, is contributed by the larger portion of the heart, which is the ventricle. So R wave is the depolarization of the ventricular cardiomyocytes. And then before the R wave, this P wave is the depolarization of the atrium cardiomyocytes. So let me repeat again. This whole page, there's the only thing you need to remember. P wave is the depolarization of the atrium cardiomyocytes, not pacemaker cells, cardiomyocytes. And R wave is the depolarization of ventricular cardiomyocytes because they are huge, they're a huge number. And T wave, according to the textbook, honestly, I've studied for years. I never figured out why it happened. According to the textbook, it said that T wave is the depolarization. No, T wave is the repolarization of the ventricle. I still cannot figure out why repolarization contribute to a positive charge of the heart. So just keep, I will not test it, but in the future you may be tested. If you test it, the typical answer is that T wave is the repolarization of the ventricle. I would not test it because I don't know, it doesn't make sense. So, but anyway, that's it. We'll have some fun lab, I think lab 4 or something. We'll talk about this, or lab 3. So we are almost done. And in the case of pacemaker, these are, we have artificial pacemakers. If you're interested, you can read. There's also different ways to increase pacemaker. There's several heart disease. And one type of heart disease is the degeneration of the pacemaker cell. And then if the heart do not have enough pacemaker cells, the heartbeat will reduce, and then in some cases, it may be very dangerous. So people need the artificial pacemaker. And so this page, I don't think I have anything to test in this page, but this one, this is the last chapter, last slice of the heart. If which is based on the heart, we already have three topics. So the last slice of the heart, let's talk about the heart transplantation for a severe end-stage heart failure. Heart transplantation is probably the only mechanical approach to save this person. However, heart transplantation is hard, and also is and one of the big things is it's very difficult to get a heart. And here in this case, I have listed three latest progress in the field of heart transplantation. The first one, the same thing we mentioned before, people can produce the heart muscle, heart cardiomyocytes from stem cells. And the thing is, in the approach that we talked about before, it's only treated for heart attack. It make the heart muscle and put in a patch and attach to the injured area. What if the entire heart has failed? How you can make an entire heart? It's not any. You cannot just use a glue or something to make a heart. So it's more difficult to reinstall the architecture of the heart. And one of the approach to combine with stem cell is this technology. You can see this heart. This is hard. This is also a heart, but different between this one and this one is that the panel B is pale. Why is pale? Because this is actually a pig heart, and then pig heart you can hardly transplant the pig heart to human because human immune system will furiously reject the pig heart, but we can really use the architecture of pig heart. In these cases, they're using a detergent to remove all the pig heart cells and only remain the scaffold of pig heart. And theoretically, people think about to inject the stem cell inside to reestablish human heart out using the pig heart structure. This is one area of the research. And so far as I know, it's really going slow. However, this is a bigger thing here. The second one, we just mentioned it used to happen before that some people spend their whole life, whole career, 20 years or 30 years trying to find the right combination of immunosuppression so that we can transplant a pig heart to a human because pig is easy to find. It's much easier to find a dead pig to donate a heart than find a dead person, deceased person to donate a heart. However, it turned out to be impossible. No matter how you adjust your immunosuppression, still a human has a strong rejection against a pig heart. So if that's the fact, why you talk about this? A huge breakthrough happened in the past 2 or 3 years. If we cannot adjust the human side, can we do something toward the pig side? Can we make a pig similar to human? That's the status. Now there's some people are working on it to modify the pig genome, to replace the pig genes with human genes. And then by modifying, sometimes, I don't remember the number. Oh, here, I wrote here, 10 genes. In this case, they replace 10 of the pig genes with human genes. And then they can transfer the pig heart into the human. It actually didn't work perfectly. The guy who transplanted pig heart, the good thing is he does not require any immunosuppression, and he lived for another 3 months. But unfortunately, this heart eventually failed for some reason. But it's a huge advantage. So we are still working on improve this. And then the key thing here is why it can happen now. That's because this technology, CRISPR-Cas9 system. I hope you can remember this one because this will be the test. You can write down on your notes. So I will definitely test this one because this is the biggest advantage applied to many different areas. It's evolutionary technology. What's the key issue about the CRISPR-Cas9 system? With this system, and now we can really edit the genome. We can edit the genome. We can edit the genome for many different reasons. We can add a genome of a pig so that pigs can have human features, can use as an organ donor. We can add a genome of the mosquitoes so that mosquito can be eliminated from the environment, from the wildness. We can modify the human stem cells so that we can generate a new treatment for sickle cell. You may have heard of sickle cell. We're going to talk about this. These are real drugs out there. 2 companies now provide a cure for sickle cell based on this technology. However, it's very expensive. We'll talk about it later. Each one single treatment costs $1 or $2 billion. No, $1 billion, $1 million to finish it. So that's the CRISPR-Cas9. Lastly, mechanically, there's another approach, another direction of this technology. What if we can replace the real heart by a mechanical heart, artificial iron heart? So this actually lasts longer than the pig heart. In the artificial heart, it's nowadays it lasts longer. The last one, the biggest one, lasts 1 to 6 years. It's amazing. And this one actually, the old artificial heart, once the people receive this heart, these people have to stay in the hospital for the whole time. But now this new one, the people can really go home with this new artificial heart, go home and waiting for the real transplantation. Now this artificial heart only given to people who have to replace the heart, but cannot find a heart donor. So this is make a buffer. So wait for 1 to 6 years. And the key thing about this heart, much better than before, is that this heart using a magnetic valve to make it last longer and the people can go home. Okay, so
Professor?
that's yes.
I'm so sorry to cut you off.
No, I'm
I
done.
have to go, and I'm not sure if you took attendance yet.
Oh, don't worry. The attendance is taken automatically in this class. So don't worry. I'm done.
Thank you.
I'm done.
Thank you.